Ferdinand tutorial. We're going to be talking about the determination of the primary structure of proteins. Now, in our last class, we've been talking about proteins. We've been talking about amino acids, polypeptides, peptides, and the classification of these proteins. Today, we're going to be talking about the determination. Now, if you've not subscribed to this channel, please do well to subscribe, like this video, comment, and share to your friends who don't know about Ferdinand tutorial. Let's run it. Now, we're talking about determination of primary structure of protein. What do we have in mind? Before we're talking about determination, what do you have in mind? Now, when you hear about determination of the primary structure of protein, this means identifying the amino acid sequence in a particular protein. Identifying what that primary protein is made up of. Are you getting it? Now, you will have to identify the amino acids that combine together to form what? To form these polypeptides. That is what? Proteins. Now, when we are talking about determination, now remember that proteins are first of all purified before they can be determined. They must be purified first before what? Determination. So now when we are talking about purification, purification simply means using various techniques. Are you getting it? Using various techniques to purify these proteins in order to make the amino acid what? Straight, in order to make it what? More cleaner for what? For what? Determination. So what do we use for this purification? Now we use the column chromatography, we use the partition chromatography, we use the paper chromatography, we use the exclusion chromatography, and many things, many other chromatographic what? Techniques. That is to say that chromatography is the main technique that we use to what? To purify this protein before determination. So that is it. Now we have these determination techniques. I'm going to be explaining only two. That is what? The Sanger's reagent, which is used to determine protein, and the what? Edmann's reagent. Now, apart from this reagent that is used to determine protein, we can also use genomes. Are you getting it? We can also use genomes. We can also determine protein according to its molecular mass. Are you getting it? We can also use according to molecular biology. We can determine protein in various ways. Are you getting it? We can determine it up to 5 or 6 ways. But I want to explain these two major ones. Are you getting it? Where we use Sanger's reagent and what? Edmann's reagent. Now, when we're talking about Sanger's reagent, how does it work? Now, remember that we have a man known as what? Frederick Sanger. Now, this Frederick Sanger uses this Sanger's reagent to determine protein. Now, how does it work? Now, Sanger's reagent contains 1-fluoro-2,4-nitrobenzene. Are you getting it? Now, this reagent, we use this reagent to determine the N-terminus of a protein. Let's run it. Let's check how it works. Now, if I have command my drawing. Now, if I have now this is, I'm trying to draw the Sanger's reagent. This is Sanger's reagent. 1-fluoro. This is Sanger's reagent. Are you getting it? Now 1-fluoro-2,4-dinitro what? Dinitrobenzene. Now with this reagent, if we react it to a particular protein, are you getting it? We can be able to determine the what? The primary structure of what? Of that protein. Now if I react this reagent with a particular protein, now when I'm reacting, there must be I'm reacting in the presence of HF, that's hydrogen fluoride. Are you getting it? That's what we work there as a what? As a catalyst. Are you getting it? That is to show us the particular protein that this protein is made up of. That the sequence, the amino acid sequence that protein is made up of. That's what Sanger's reagent is talking about. Now remember that this Sanger's reagent, it doesn't only start from determining from the C-terminus. No, it must start determining from the what? From the N-terminus. That's the one this side, the one that will begin. That's N-terminus. That's this one now. H2N. Are you getting where we have C? We have C-O-R. We have R here. We have here H. This is a structure. This is a sketch of a protein. This is the protein here. Are you getting it? Now, if I'm reacting with this protein, now let's say that I will have in the presence of HF. Now, if I have HF here now, you see that my protein now I will form a new protein now, which is this. I will have have, are you getting it? Now I will have, now remember this F have been limited. This F is here, one hydrogen is here. Now what do we have here? We have only H here, we have only N here, H, we have C, we have R, we have H, we have CO, R. Are you getting it? Do you understand what I'm saying? So that is it here. Now we formed another new protein. Are you getting it? We formed another new protein. Are you getting it? So that is it for what? That is it for Sanger's reagent. It only determines, it only start determining from the what? From the N terminus. From the N terminus, which is here. Now from here we can determine, we can know the protein that is here now. From here we can determine the protein that is here. So that is all for what? Sanger's reagent. Now we are talking about Edman's reagent. Edman's reagent, yes, it only starts from what? It only starts from this what? N terminus also. Now Edman's reagent is only used to identify one protein structure, because this reagent reacts with many proteins. Are you getting it? It is only used, it can only be used at a time to determine each what structure, that is each amino acid sequence in a protein, what that protein is made up of. Are you getting it? We can only use it once only. Are you getting it? So that is it for Edman's reagent. Now, if we have a structure like this, if you have a structure like this, are you getting it? And maybe we have something like this, and let's see, yes, and we have something like this. Are you getting it? Now with this is a sketch. Are you getting it? This is a sketch of what the Edman's reagent is all about. Now with this, now we can use it to determine the N terminus of a protein. Also, remember we only determine one structure, one particular structure of that protein. Now when this reagent, Edman's reagent, reacts with a particular protein sequence, now this place now fix itself. This reagent fix itself to the N terminus, thereby making us to determine what? Thereby making us to determine the structure of such what? Protein. We also have molecular mass of a protein. That means when amino acids combine together, now they have a varying molecular mass. Are you getting me? At each point, they have been able to determine that particular, at each level, they have been able to determine the particular amino acid present there. Are you getting it? So that is all about this what? That is all about this class. Now also, not only this reagent, there are many other reagents, as I explained earlier. There are many other reagents apart from these two agents that I explained here. So, but these two main ones are the one that we use in our daily lives. So that is it for this class. That is it for primary structure of protein. I'll be talking about plasma proteins in my next class. So try to be present. Try to hook up. Try to tell your friends about it. The class will be interesting. Please like this video. Please like this video. Comment and share to your friends. Thank you.